Welcome back to this week's OIS podcast. Today, our host, Dr. Rob Rothman, speaks with Stuart Therapeutics President and CEO Eric Schlumpf about how he and his business partner discovered a family of collagen peptides that have led to a novel anti-inflammatory approach to dry eye disease. Take it away, Rob.
Hello, OIS podcast audience. It's a pleasure to be speaking with you again. For those of you who do not know me, my name is Rob Rothman. I am the co-founder and co-managing member of InFocus Capital Partners, which is an ophthalmic-focused venture capital fund. We have currently completed our investment cycle for Fund 1, closing with 13 assets, all focused on different areas inside of ophthalmology. I spend some part of my working hours as a clinically practicing ophthalmologist. I'm a glaucoma specialist by training. So I have the ability to hopefully speak with some interesting people regarding their ophthalmic opportunities and understand those opportunities from both the perspective of a clinician and an investor. Having said that, it is my absolute pleasure today to be speaking with Eric Schlumpf, who is the President and CEO of Stuart Therapeutics. For full disclosure, InFocus has invested in Stuart Therapeutics, one of our first investments actually, early on in our cycle. And I believe that some of the recent developments regarding Stuart's progress as both a business and an ophthalmic asset have accelerated to the point where it probably is a good idea for Eric to have the chance to present some of that information to the listening audience. So Eric, thank you for taking the time to be with us today.
Appreciate it.
I can see from your background that you have similar weather to me, except it's probably about 100 degrees hotter where you are than I am.
Maybe not 100, but it's warmer, definitely.
Yeah, and the humidity is probably a little higher too. So that's
Indeed.
Too bad. So let me first get this going by, I've had an opportunity to know you for several years now during the whole due diligence process from InFocus. And, over the last couple of years since we've invested in trying to help Stuart progress. So I think that one of the reasons why I was interested in having you as a guest is because I think your background and the story as to how you got to where you are is very compelling. So, maybe you could just start by taking us through Eric Schlumpf's journey from, a small boy all the way through president and CEO of Stuart. And obviously I think understanding your background from both where you grew up and educationally is important, but mostly through your work experience and how it led you to Stuart. So fire away, and I will sort of, direct you towards those things I think are super interesting when we get to them.
Great. Thanks, Rob. And appreciate the opportunity to talk with you today. So I got started as an engineer. I'm an electrical engineer by training, and then got bored with that very quickly and decided to get into business and got a master's in business and did some merger and acquisition work for a handful of years as I was getting started. And ultimately I started to gravitate towards startups and early stage companies. And even when I've worked in large organizations, I've been doing the new thing or the new venture within a large organization. Had a turn in the waste management industry, got involved in management consulting, started a management consulting firm that was quite successful and got sold. And I ran a big piece of T-Mobile's business for a while. So I've kind of bounced around a little bit in telecommunications and so forth. But a big chunk of my advisory services work was in advising large pharma companies and some medium-sized companies as well on product development activities as well as supply chain issues. And that got me exposed to pharma and interested in that side of the world. And as I was involved in some businesses in Southern California, I got to know my current business partner, Bob Barada, who's our chairman. And Bob and I just hit it off. He's a longtime ophthalmologist here in South Florida, just a tremendous guy, very, very sharp business guy. Sort of the antithesis to all the jokes about doctors not being great business people. Bob's a fabulous businessman. And Bob and I got to know each other, got involved in some businesses related to ovine collagen and wanted to do some work in that. And as we were researching those business opportunities, we got a hold of some technology that we found at the University of Wisconsin. And the inventor, a guy by the name of Ron Raines, who's now at MIT, had come up with an idea to synthesize a fragment of type 1 collagen. So it's a synthetic fragment. And he designed it so that it would bind to damaged collagen triple helices and heal them very, very rapidly. And as we began to talk to Ron about the applications of this technology, we got really, really excited about it. It seemed like a white space that no one else was looking at. It seemed like something that had applicability in a large number of different diseases, inflammatory diseases, where matrix metalloproteinases are digesting collagen and creating an inflammatory cycle. So we grabbed a license to the technology from Wisconsin, and we spent about 6 weeks arriving at what turns out to be the inevitable conclusion that we were going to be in ophthalmology, with Bob being an ophthalmologist. And we started Stuart Therapeutics, and our first series of tests that we ran were on corneal wound models in mice. And we found that we had a dramatic healing effect on damage to corneal tissues. And then we've just expanded since then. We've done additional testing to get a deeper dive into the mechanism of action. We've carried the initial corneal wound testing into an application and a drug candidate for dry eye disease, which is now in phase 2 clinical trials. And we're right behind it with 2 additional indications, one for neuroprotection, the optic nerve, and glaucoma. And then the final one we think we've got real possibilities in is dry age-related macular degeneration. So all of that journey took about 3.5 to 4 years. It's been good. We've appreciated the support of those that have helped us along the way, including In Focus, been tremendous partners for us, as well as the University of Wisconsin folks at the Alumni Research Foundation, who've been good licensed partners as well as investors in the company. So it's been a great journey. We've had a great time with it so far, and we're in a good place right now. We think things are moving along nicely.
Okay, we're going to get to all that, but we have to go back a few steps, because I think that there's some missing Darwinian links there in the evolution of you. Right. For example, waste management. Telecom, right? So waste management, telecom. So just thinking about the skill set that you have, and again, with the caveat that I know you fairly well, how does that occur? I mean, is that just a job shift? Is that just there are lots of people who listen to these podcasts from all different walks of life, and some of them might be people who are wondering, how do you become the CEO of a startup ophthalmology company?
Yeah,
so
right.
And we're going to get to that comment, which is the third missing link, which is bovine collagen, which you just threw in there.
Right,
and, it's not common to have a waste management guy throwing around terms like matrix metalloproteinase and all these other cool things. So so how do you go from somebody involved in, let's say, a service industry to another service industry to eventually into, biotech at the startup level, because those are completely different worlds?
Yeah, I think there are a couple of different dynamics that have gone on. I think one of the things that I wanted to do early in my career was get out of being a staff guy working at a corporation. So the first thing I did was to desperately try to find a new area of business that I could go off and lead. And that, the first one that lined up was the waste business. And I got into medical waste because I wrote a business plan for the CEO of the company that I was working for. And I happened to turn it in on the Friday before the New York Times articles came out about all the med waste rolling up on the shores of Long Island many years ago, unfortunately. So my timing was perfect. And so I got handed that opportunity, and I got involved in the waste industry in that way. And I ran that string out for a while, and it led me into startups and how to deal with young organizations and how to build staffs and make sure that you don't make the mistake of not deciding on things, which I think is critical in startup companies. So that was one dynamic. And I think that led me to be creative and interested in doing new things. Maybe that's the I get bored easy dynamic in my life. But it also forced me to not get outworked. I always want to get into something, and I want to know it, and I want to understand it so that I can communicate it effectively to people who are interested in it. So that's another thing that's been a constant through my career. Beyond that, when you get into the other elements of my career, a lot of it was built off of being a partner in management consulting firms, because you're exposed to senior executives. You've got to be on your game when you go in to meet with them, and you got to know your stuff about their industry, and you don't have a lot of time to prepare for it. So it forces you to prioritize knowledge and prioritize how you approach problem solving. And I think that's a secret to being successful in any small venture, just as it is being successful in advising companies about their business. So you can't be credible unless you are able to make that prioritization happen. So that's kind of got me into a number of these situations. The telecommunications thing came about because of a friend, and T-Mobile was going through a bunch of major changes, and they needed someone to run one of the largest business units for them. And I just hit it off with the CEO at that time, and had a great time working for that organization, learned a lot about large companies and large numbers of customers and statistics in the process. But I think as I kind of got involved in life sciences off and on in my life, laboratory services, pathology laboratories, and management consulting to major corporations, what always intrigued me was the opportunity to try to help folks to solve health problems. And, the situation with ophthalmology is a close one to me. My folks both have problems with their eyes, and being able to provide solutions that are going to ultimately help them was something that was important to me. So that kind of drove me in this direction in a big way.
So how did you first meet Bob, and what was the impetus for you to decide that, hey, maybe I'm going to leave my, highly compensated management role with a large, company and try and slog it out in the trenches of startup ophthalmology corporate life? I mean, how did that happen?
Yeah.
What was the dynamic between you and Bob at that time, and how did that get started?
Yeah, Bob and I got introduced by a friend of a friend. I came out to visit him here in Florida. My wife's family were getting into end of life challenges, and it started to become smart for us to think about being here in Florida to help them out, to deal with some of that burden that her sister had been carrying for a number of years. So when Bob and I talked and I got to know him a little bit better, I just found a creative guy, kind of a kindred spirit in terms of somebody that's interested in trying and doing new things, and somebody who had a brilliant background of his own. And I think that makes a difference. I mean, you don't run into people like that often in your life, where you feel like you can work together and you feel like you can work effectively together, who you also enjoy being around. And you need to grab those opportunities as much as you can. One of my other colleagues who I've worked with for a long, long time always says you can do a lot of work, but it's always a whole lot better when you can do it with people that you enjoy being around. So we hit it off very well. Our interests are aligned very nicely, and we're pretty complementary. I'm kind of the commercial and business development and financial oriented kind of guy, and Bob's great at the patient and physician and clinical aspects of things. And when you surround yourself with our other business partners, which include folks like InFocus, of course, the folks that are our CRO partners at Aura, and so forth, it really makes for a great combination.
Yeah, that's great. I mean, again, I'm a little bit biased because I know the path and the story, but yeah, I agree. Obviously you have to have people surrounding you who are complementary and pleasurable to be with. One of the valuable lessons that we've learned in the investment cycle, part of our business and doing diligence is the first three check boxes for any company are diligent, management.
Yep.
Right. If management isn't check, then it doesn't really matter for the most part how good the asset is. It's just going to be very difficult to get anything to move forward. So
yeah, indeed.
So yeah, we would agree that, you found some great people to work with you, and that's most definitely helped propagate you. How, just in a little bit more detail, how'd you find PolyCol? Because again, as an investor in a company, we're fascinated with that product and we think that it's going to be incredibly successful. But how did you really find it? I mean, you hear these stories all the time about, assets or compounds or chemicals or whatever they are sitting in some, portfolio
Dusty library.
And somebody finds it and realizes there's an opportunity and a potential, and they dust it off and they turn it into something incredibly amazing. So how does that happen? How did that become apparent to you, and where did you come across it, and how did you get involved with the Wisconsin people?
Yeah, it was when Bob and I, we were engaged in what really amounted to a roll-up of wound care companies for a variety of different reasons that don't really matter to the PolyCol story. And one of the challenges you run into with bovine collagen is mad cow disease. And there's always that sort of specter lurking in the background that somebody is going to end up with a zoonotic indication because they use a product that emanated from cattle. And it pops up often enough that it's kind of the thing you look over your shoulder about in that industry. So we had decided to look into and research if there were any synthetic opportunities in collagen that were being researched. And there are really only 3, maybe 4 folks in the United States at that time who'd been doing any work at all in synthesized collagen. And it was all focused in a variety of different directions that didn't seem to matter too much. And then we found Ron at Wisconsin, and his technology caught our attention because he had done some wound healing tests in mice with it. And we saw dramatic results in animal skin wounds. And because we'd been in the burn area and the wound business a little bit and knew a little bit about that, it really raised our eyebrows. And we just kind of on a whim decided we were going to zip up to Madison and sit down with Ron. We set an appointment with him, and we spent a day with him and talked to him about what he had in mind and what he did. And, a lot of his conversation was over my head because he's just one of the most brilliant chemists in the country, very well-known guy. But as we began to talk about it and began to think about the application of it and where it could go in medicine, Bob and I got very excited about it. So as we left, we contacted the IP office there at Wisconsin, at the Wisconsin Alumni Research Foundation, and asked them if we could negotiate an option on the license, and which we did. And then we took some time to figure out what we wanted to do with it. And the mechanism of action of this thing is pretty interesting. It binds to the portion of the triple helix in collagen, triple helix domain, that is unraveled by collagenases, matrix metalloproteinase. And that unraveling creates a structural dysfunction. The cells that depend upon collagen can't effectively heal or propagate. And it also creates a cell signaling dynamic that's pretty important. It's contributory. Once it's unraveled, it contributes to inflammation, and it ceases to offer the cell propagation and proliferation signaling that is normal tissue homeostasis. So when we looked at that and we started to think about where we could take that, the eye was an obvious place for us to go. There's a lot of collagen in the eye. There's a lot of opportunity in the eye. And we felt like it would be potentially an option for a number of different inflammatory indications, chronic indications for which there aren't great satisfactory solutions. So that's kind of how we centered on that. That's how we got it rolled in. We branded it as PolyCol. We have the rights to a number of sequences from the University of Wisconsin, each one unique. We utilize different ones for the different disease states that we're pursuing, disease indications, And we've also filed additional intellectual property with some new sequences of our own design and additional use applications as well. So it's been a nicely expanding intellectual property portfolio. It seems to be fast and effective. As I mentioned, I think at the beginning, it's kind of a white space product. There's nobody else really looking at this kind of thing right now. And it gives us a free field in ophthalmology for indications that we find important. And we hope to have a great impact.
Yes. So there's a couple of questions that I think would be important for the audience to understand. And I think they should hear from you in terms of why you think this is important. I don't have to tell you, and I certainly don't have to tell most of the people who listen to these, that, better therapy for dry eye is sort of a key component of almost every investor in ophthalmology. Everybody's looking for something better than what's currently available. And there are a multitude of reasons why the currently approved non-OTC remedies for dry eye have been sort of relatively lackluster in their performance, despite the fact that they continue to generate significant, 10-digit sales.
Right.
Despite the fact that if you ask almost every clinician out there, they're not great.
Right.
So why do you think, why is PolyCol going to be something that's better than what's out there? And why do we think it's unique? And, how does it stand out against other potential products that are currently in development in broad terms, I guess?
Yeah. I think the two biggest elements of our value proposition in dry eye disease, and I agree with you, by the way, that it's one of those things where everybody hates the result, but they're still spending money on it, is that we work very quickly. The results that we've gotten in animal models have been 24-hour results to a complete healing in fairly severe corneal wounds. So speed of action is important. And I think that's the big missing element in almost every therapeutic in dry eye disease right now. And then the other element of this is that the breadth of applicability. So the narrowness of some of the therapies, brand name therapies that are out there and being prescribed right now is such that it's a little bit of a crapshoot, if you'll pardon the term, for patients when they go into the doctor's office as to whether the therapeutic is going to work for them or not. We see collagen damage in almost all dry eye disease patients. There's extracellular matrix collagen inside of the epithelial layer in the eye. There's Bowman's layer and the stromal tissues that underlie that. And everyone knows that matrix metalloproteinase levels are elevated in dry eye disease patients. And that substance has one role in life, and that's to act as a collagenase and damage collagen. And it does it by opening up those triple helix domains in collagen in the eye. So we see a wide open field for us. We see ourselves as being very broadly applicable and working very quickly. The other element that we've seen, and this has been some in vitro work that we've done more recently, is in the restoration of the cell signaling role of the triple helix collagen. Once we do our repair, we're having an impact on inflammation that's pretty dramatic. The in vitro testing that we've done on IL-6 and IL-8 expression by epithelial cells shows us returning within 16 hours or less the IL-6 and IL-8 levels to naive. And that's suggestive that this mechanism of action is a very robust one. And we're excited about the ramifications that has for ocular surface indications, as well as indications in other parts of the eye.
Yeah. So, and again, just to simplify that, I think for people who aren't as familiar with the whole disease process, but dry eye is a multifactorial disease, right? There are loads of different pathways of inflammation that may be at play here. There's multiple different causes. And one of the concerns that we've had, for example, as an investor has been, well, how do we know if we invest in your novel anti-inflammatory approach towards dry eye, that's the pathway that's most important?
Right.
Rather, you are focusing on the end product, which is the actual damage to the surface epithelium of the cornea. And if you can repair the corneal epithelium, then you're going to decrease the signaling for inflammation that causes all the symptoms of dry eye from the reverse way. So it's sort of a reverse approach towards dry eye, which is let's just fix the problem and everything else goes away. It makes sense. If you close the wound and you heal the wound, you're not going to have anything asking for an inflammatory process, right? I mean, that's the way, that's the simplified way that we looked at it is, you're trying to fix the damage. Forget trying to, figure out where the termites are living underground. Let's just actually make a house that you can't burn down.
That's exactly it. And I think being agnostic to the cause or the etiology of the disease is actually an advantage for us. We don't really care whether you've got a meibomian gland dysfunction or a tear film challenge, composition challenge, or if you've got exposure issues or what have you. We're going after the causal damage in the tissues that's starting this, as it's described in the literature, vicious cycle of inflammation. Right. And so we think that's a winning formula.
So how did the FDA handle this? This is something new, right? This was not typical for the FDA, I believe, in terms of evaluating dry eye. So how was that process? I know that you sort of seemed to breeze through it a little bit. So how did it go? I mean, what was their response? I mean, I know you're in the middle of your phase 2 clinical trial, so you can discuss that a little bit if you want, but I think the FDA was fairly receptive to this.
I think part of the benefit that we had is that the molecule is an approximate mimic of a fractional strand of type 1 collagen. So it's got the same amino acid sequences in the similar kinds of order that you normally see in collagen. And I think when we spoke with the agency, they looked at that and they said, okay, this looks pretty benign. If you can come to us and show us in your preclinical work that you've got 2 species, toxin pharmacokinetics that are squared away, then, and I'm paraphrasing here, I don't think the FDA ever says squared away, but, if you can come to us with that data, then we'll allow you to move directly into a phase 2. And that was attractive to us because we wanted to get the technology into a phase 2 trial as quickly as we possibly could, is shorten the development cycle and not have to go through several months of, the phase 1 exercise. So we took that path. We got all of the data. The toxin pharmacokinetics work was completely clean. We expected that, but it's always nice to get that confirmation in our industry. So we got that, and we submitted the IND earlier this year, and it turned around with a let's go get it done. And we've got a phase 2 underway right now that we expect to have top line results in October.
So formulation, CMC, all that stuff, relatively quick. I mean, this is really just a vehicle for delivery more than anything else.
Yeah,
right?
it's the formulation itself is saline with a minor tweak to pH and a little bit of an osmolarity change, so it's not too much different than saline. The peptide itself is manufactured as a salt, a dry salt that's then dissolved in solution. It appears to be quite stable. So a lot of the unknowns about the development of this as a marketable drug substance were knocked out during the CMC process for us, and we're very fortunate that went well. I'd say in terms of how quick it went, it was frustratingly slow from my perspective. Last year was kind of a nightmare for a lot of reasons, but we got through it okay. We stayed more or less on schedule. We had a couple of minor delays that slowed us down, unfortunately, but being able to go into the first quarter and being able to get the IND submitted in the first quarter of this year was about when we wanted to get it done. So we're pleased with where we sit right now.
And it's a pretty straightforward study, right? So it's a fairly straightforward study.
That's
not
right.
very long, not a whole lot of patients required, and we expect that you will have data sometime to discuss.
Yeah, October top line, and hopefully November for the full report out. It's just kind of a standard dry eye clinical trial, about 150 patients. We got 2 different dose levels and 1 control cohort in the trial itself.
That's fantastic. Mm-hmm. So we got the dry eye. Now I think that it's really compelling to discuss at this point some of the other potential applications. We view, and I think you do, clearly view PolyCol's platform technology.
Right.
There are other applications here because we are fairly certain, I think, as scientists, that collagen damage has a role in every disease state. Clearly, there's focus now on glaucoma potentially having some impact from collagen damage at the level of the lamina cribrosa, or even ganglion cell structuring complexes that contain collagen. And then clearly, obviously, the known issues with collagen damage in AMD, I think, to some degree. So how does that all play into where you're going to go with this moving forward? Because there are other indications to pursue here, right? I mean, Sure. It's not common to see platform technology, and let's forget the fact that they have to get this product to the target tissue, and we're not going to talk about delivery or anything like that now. Just theoretically, how do you foresee PolyCol interacting with other disease states in the eye?
Yeah, I think the one that is probably next in line for us is Neuroprotection for glaucoma. And our chief science officer, David Calkins, who's also at Vanderbilt University, and this is his field. And from the day he got connected with us, he was bugging me to allow him to run a series of tests on this compound or a related sequence in the family of compounds, if you will, in some models that he had of glaucoma. And when we were able to afford those tests, we ran them. And what we found was a pretty remarkable degree of protection of the retinal signal to the portion of the brain that processes that. David's model elevates interocular pressure, and he allows a damage cycle to occur, and he does it in both rodents as well as primates. And then he has a unique way of measuring signal transport from the retina to the brain by using a dye that travels up the optic nerve. And when we ran those tests, we found that PolyCol, a PolyCol variant, was incredibly protective of vision transport, which is one of the earliest issues in glaucoma. It starts to drop out early. The actual nerve fiber layer dropout happens much later in the process. So those effects are important. And when we ran those tests, he called me very excitedly and said, boy, he does these tests for all the major pharma companies for neuroprotective compounds. He said, this is as good or better as anything I've seen, and it has apparently none of the off-target effects. So that combination of apparent safety and effectiveness is profound in this particular opportunity. Doesn't mean it's not going to be complicated. Neuroprotection's got a storied history in clinical trials, and we're going to have to figure out a way to get that done economically and get our head together with the FDA, which we're hoping to do this fall, about what the clinical trial might look like and what those endpoints are. But, I always say that, if somebody's going to do it, it might as well be us. So let's figure out a way to get that done and get something out there that's really going to help people who struggle with a disease that still blinds an awful lot of people every year. And, it's near and dear to my heart. My dad suffers from some pretty severe glaucoma challenges, and I'd love to be able to have something where I can knock that out and attack that problem in a big way.
Yeah, it'll be interesting. I think we're excited to see that as well. And obviously, I think the same concept applies towards the AMD pathway down the road. I think it'll be great to see how you can navigate the pathway of trying to either prove that this drug has a benefit, or at least get some indication that that's the case, and then subsequent funding to develop those products. In some way.
With AMD, it's interesting because you've got where we work in the front of the eye with Bowman's layer and the stromal tissues supporting the corneal epithelium. You've got a corollary in the back of the eye in the retina with the retinal pigment epithelial monolayer supported by Bruch's membrane. And we know there's damage back there. We know that it's disrupted in dry AMD. And our feeling was that we could apply in a similar way this product to the back of the eye and create some healing to Bruch's membrane and a more friendly environment for RPE recovery, if we can be as bold to say that. There's also extracellular matrix collagen in and around the ganglion cell layer that we know about, and the nerve fiber layer. And we think that there's applicability there as well, but we got a lot more work to do, I think, on the AMD side of things, just in terms of proof points and understanding the details of the mechanism of action and how it applies there. We're not quite as far along there as we are in neuroprotection.
Right. Well, dry eye is clearly the low-hanging fruit for you. That's
indeed.
the easiest validator that you have, and obviously presents an opportunity for significant return to the company in terms of its ability to propagate itself into other areas. Obviously, this secure potential future investment from people who are specifically interested in things other than dry eye. So I think the opportunity is there for you. And, based on the results of the dry eye study, it'll be interesting to see what happens after that. You've raised a pretty significant amount of money, which is great, partially thanks to
Indeed.
But clearly
thank you very much.
But clearly not an easy task. And, raising funds over the past 2 years is a little bit of a challenge. I know firsthand for you. So that went, you achieved your funding target, correct? So you raised how much?
Well, we raised, we put a total of about 12.7 into the company. The A round was an 11 million round. We started off with a $6 million Series A round, and then a number of things happened, including COVID last year, which slowed things down. And as we kind of got towards the end of last year, we sat down and took a hard look in the mirror and said, look, we want to push this forward. We want to get this thing into a phase 2 trial. And we think the right strategy is to bump the round from 6 to 11, take a little bit more dilution. And we've made more progress, and we've got more to talk about now at that time than we did before COVID kicked in. And let's just go ahead and increase the round, and let's push the pedal to the floor and try to get it in so that we can get the IND filed in the first quarter. I'm happy where we're at right now. I keep thinking back to the weeks in March of 2020, where we had a couple of venture capital firms, small, medium venture capital firms that were ready to sign up with us. And then COVID started looking bad, and they said, we need to retreat around our portfolio. So we sort of swallowed hard and said, okay, we'll figure out a way through this. And ultimately we did. I think it's always hard to raise funds to support something that's new and different, which we represent. It's easy for people to say yes to even gene therapy nowadays, right? But we represented something that required a little bit more explanation. And hats off to the InFocus folks and the other folks that have been confident in us and invested in the company, because I think they had the vision to see that this could be something different that would be fundamental and important. The process was tough to get through, but rewarding nonetheless.
Yeah, I would agree. And then, so what about future funding needs? You're contemplating raising some additional capital now
Yeah, we're raising a convertible note right now, a relatively small one. We want to push both the dry AMD and the neuroprotection assets as quickly as we can to where we can be a real viable 3 indication entity next year. We've got we'll have some decisions to make about what we do with the dry eye asset, depending upon the results of the phase 2 trial. There's interest, I think, in the outcome there. I've got a long list of people that say, come see me when you got phase 2 results. So that's probably not terribly unique, but we see real strong interest in the platform beginning to form, and we see opportunities in adjacent areas. The dry eye product, because of our mechanism of action, is something that could be applicable across a wide array of ocular surface indications for which there are no real good solutions. Things like corneal ulcers and recurrent erosions come to mind, and post-surgical applications as well as prep for measurements for cataract surgery, starting to become an issue with folks, anterior segment folks. The other opportunities, include things like solutions to support extending the life of a wearer for a contact lens company. If we were able to come up with a variant that would be packed with contact lenses, and that just means instead of putting them on the shelf at age 50, you put them on the shelf at age 55 or 60. So that could be a good value proposition as well. So we're looking at all of those things. We think there's great opportunity in those adjacent areas, and the conversations are starting to become the nicer kind of conversations you like to have, as opposed to the ones where you're trying to punch through the door.
Yeah, listen, it's a fascinating approach, and again, I think you hit it on the head that I believe the group of people who have supported the company at this time are those who are interested in seeing something new and unique and potentially disruptive into the market for dry eye that is not another version of cyclosporin.
Yep,
right? Which has always been the bane of most, dry eye specialists or ophthalmologists is, I have cyclosporin A, cyclosporin B, and cyclosporin C for you to try, and artificial tears, and maybe this will be better. So I think that everybody, and I've had conversations with a lot of those investors, over the course of the last 2 years, obviously, and they all seem to have the same mindset, which is that this seems so elegantly simple and so logically sound that it's a viable place to try and invest. So congratulations on identifying that and propagating it. It has not been an easy time. You chose to pursue the clinical pathway probably at the worst time in history, at least
exactly
in the last certain amount of years, and managed to fund the company, get a clinical trial approved and initiated to the point where you will have top line data within the next 2 to 3 months. So I think with that as sort of a final sentiment, I obviously want to congratulate you.
Thank you.
I'm looking forward to seeing what the data looks like and seeing how the company continues to progress from being a dry eye company to being a platform company that may potentially address things as important as glaucoma and macular degeneration as well. So for the audience, I will say thank you. We obviously all wish you much success. Again, Eric Schlumpf, President and CEO of Stuart Therapeutics. Pay attention to this company. We think it's going to be a big one. So thanks everybody for listening in, and I'm looking forward to future podcasts with you down the road.
Thanks, Rob. Take care.
Take care.
Thank you for listening, everyone. We hope you've enjoyed this episode of the OIS podcast. Be sure to subscribe to our iTunes channel so you don't miss any ophthalmology insights. Got a story of your own to tell? Apply to be a guest at OIS.net.